Good morning, dear friends. Myself, Abdullah Sherikar, working as an assistant professor in the Department of Pharmacology at Sri Vile Parle Kelavani Mandal's Institute of Pharmacy, Dhule. Today, we are going to study about blood, that is part 4, that is red blood cells. Learning outcomes: after completion of this lectures or video lecture, the learner will be able to understand the detailed physiology of the RBC, detailed functions of the RBCs, and the life cycle of the RBC. The red blood cells or erythrocyte contains oxygen carrying proteins, that is hemoglobin, which is a pigment that gives the red color to the RBC. A healthy adult male contains about 5.4 million RBC cells per microliter of the blood, whereas healthy adult female contains on an average 4.8 million RBCs per microliter of the blood. To maintain the normal function of the RBCs, the new mature cell must enter the circulation at an astonishing rate of at least 2 million per second. Now we will see normal hematocrit hemoglobin. The red blood cells have ability to concentrate the hemoglobin in the cell fluid up to about 34 gram in each 100 milliliters of the cells. That is considered as a metabolic limit of cells hemoglobin forming mechanism or the process. At normal hematocrit hemoglobin, the whole blood of men contains 50 grams of hemoglobin per 100 milliliters of the cell, whereas whole blood of the women contains 14 gram of hemoglobin per 100 milliliters of the cells. Now we will see the RBC anatomy. The RBCs have biconcave discs with a diameter of 7 to 8 meters, thickness 1 to 2.5 micrometer. The mature or red blood cells have simple structure. The plasma membrane of the RBC is strong and flexible, which allows them to deform without rupture as they squeeze between narrow capillaries. The RBC also contains certain glycolipids on their plasma membrane that are called as the antigens that account for the various blood groups such as ABO and RH system. RBC constitute about 33% of cells weight. Next, RBC physiology. RBC have highly specialized oxygen transport function. Because RBC have no nucleus, all their internal space is available for the oxygen transport. As they lack mitochondria, they generate the ATP anaerobically, that is without utilization of the oxygen. Therefore, they do not use up any of the oxygen they transport. Actually, the RBC is a bag that can be deformed into almost any shape. Even the shape of the RBC facilitates its function. Therefore, a biconcave disc has a much greater surface area for the diffusion of gas molecules in and out of the RBCs. Each RBC contains 280 million hemoglobin molecules. Now next, functions of the RBCs. A hemoglobin molecule contains a protein called as the globin, which is composed of the 4 polypeptide chains, that is 2 alpha and 2 beta chains, whereas a ring-like non-protein pigment called as the heme, which is bound to each of the 4 chains. At the center of each heme ring, there is an iron, that is Fe plus 2, that can combine reversibly with one oxygen molecule. Therefore, each hemoglobin molecule bind four oxygen molecules. Each oxygen molecules pick up from the lungs is bound to an ion. The RBC releases oxygen which diffuse first into the interstitial fluid and then into the cells. Hemoglobin also transport 23% of the total carbon dioxide, that is considered as a waste product of the metabolism. The remaining carbon dioxide is dissolved in plasma or carried as a bicarbonate ions. The blood flowing through tissue capillaries pick up the carbon dioxide, some of which combine with the amino acids in the globin part of the hemoglobin. As blood flows through the lungs, the carbon dioxide released from the hemoglobin and then exhaled out of the body. The hemoglobin also play a crucial role in the regulation of the blood flow and blood pressure. The gaseous hormone nitric oxide, that is NO, produced by the endothelial cells that line up the blood vessels, bind to the hemoglobin. Under some circumstances, hemoglobin releases nitric oxide. The released nitric oxide causes the vasodilation, that is, an increase in the blood vessel diameter. That occurs when the smooth muscle in the vessel wall relaxes, which increases the blood vessel diameter, and ultimately improve the blood flow. All these changes bring about the enhancement in the oxygen delivery to the cell. The red blood cells also contains the enzyme, that is carbonic anhydrase, that is CA, which catalyze the conversion of the CO2, that is carbon dioxide, and water, that is H2O, into the carbonic acid, that is H2CO3, which is reversibly converted into the H plus ion, that is hydrogen ion, and HCO3 minus ion, that is bicarbonate ion. The significance of this reaction has uh, two significances. First, it allows the 70% of the CO2 is transported into the blood plasma from the tissue cells to the lungs in the form of the bicarbonate ion. And second thing, this reaction is served as an important buffer in the extracellular fluid. Now next, RBC life cycle. First, life of RBC, that is red blood cells, is 120 days because of the wear and tear their plasma membrane undergo as they squeeze through the blood capillaries. Second point, the RBC has no nucleus and other organelles. Therefore, the RBC cannot synthesize new component to replace the damaged ones. Third point, the plasma membrane of the RBC is fragile with age, and cells burst during the squeeze through narrow channels in the spleen. Fourth, the ruptured red blood cells are removed from the circulation and destroyed by the fixed phagocytic macrophages in the spleen and the liver. Last point, that is the breakdown products are recycled, which are used in the numerous metabolic process, say example formation of the new red blood cells. Now the recycling occur as when macrophages in the spleen, liver and bone marrow phagocyte the ruptured and worn out red blood cells. This causes splitting of the globin and heme portion of the hemoglobin. The iron is removed from the heme portion in the form of Fe plus 3, which associate with the plasma protein, that is a transferrin, in the bloodstream, that is which is considered as a transporter for the Fe plus 3. In muscle fiber, liver cells and macrophages of spleen and liver, Fe plus 3 detaches from the transferrin and attaches to the iron storage protein called as the ferritin. On release from the storage site or absorption from the GIT, Fe plus 3 reattaches to the transferrin. Then Fe plus 3 transferrin complex is then carried to the red bone marrow, where RBC precursor cells take up with through the receptor mediated endocytosis for use in the hemoglobin synthesis. In this case, iron is needed for the heme portion of the hemoglobin molecule and amino acids are needed for the globin portion. Whereas vitamin B12 is also needed for the synthesis of hemoglobin. When erythropoiesis is that is the synthesis of red RBC in red bone marrow result in the production of the red blood cells which enter the circulation. Now globin is broken down into the amino acids which can be reused to synthesize other proteins. When iron is removed from the bone marrow, when iron is removed from the heme, the non-iron portion of the heme is converted into the biliverdin, that is a green pigment, and then this biliverdin is converted into the bilirubin, that is considered as a yellow-orange pigment. After this, bilirubin enters the blood and is transported to the liver. Within the liver, bilirubin is released by the cells into the bile, which passes into the small intestine, then into the large intestine. In the large intestine, in presence of the bacteria convert the bilirubin into the urobilinogen. Some urobilinogen is absorbed back into the blood, which is converted into a yellow pigment called urobilin, which is excreted via kidney in the form of urine. Most urobilinogen is eliminated in the fecus in the form of brown pigment that is called as the stercobilin, which gives fecus its characteristic color. Now the production of the RBC. Erythropoiesis, that is nothing but the production of the RBCs which start in the red bone marrow with a precursor cell called as the proerythroblast. The proerythroblast divide several times, producing the cells that begin to synthesize the hemoglobin. Ultimately, a cell near the end of the development sequence eject the nucleus and become a reticulocytes. The loss of the nucleus causes the center of the cell to indent, producing the red blood cells with a distinctive biconcave shape. The reticulocyte retains some mitochondria, ribosomes, and endoplasmic reticulum. They passes from the red bone marrow into the bloodstream by squeezing between the endothelial cells of the blood capillaries. The reticulocyte develop into the mature red blood cells within 1 to 2 days after their release from the red bone marrow. The oxygen delivery may also fall due to the anemia, which has many causes like lack of the iron, lack of the vitamin B12, and certain lack of the certain amino acids. All these causes the anemia. The cellular oxygen deficiency called as the hypoxia may occur if too little oxygen enters the blood. For example, the lower oxygen content of air at high altitude reduces the amount of the oxygen in the blood. Secondly, the circulatory problems that reduce the blood to tissues may also reduce the oxygen delivery to the blood. The cellular oxygen deficiency called as hypoxia may occur if too little oxygen enters the blood. Whatever the cause, hypoxia stimulates the kidney to step up the release of the erythropoietin, which speed up the development of proerythroblast into the reticulocytes in the red bone marrow. If these the reticulocyte after this there is an increased population of the RBC in the circulation and the more oxygen will be provided to the tissue. If the oxygen carrying capacity of the blood falls because erythropoiesis is not keeping up with the RBC destruction, a negative feedback system step up the RBC production. Next point, why premature newborn exhibit anemia? During the first week after birth, the liver produces the most of the erythropoietin because liver is the less sensitive than kidney to hypoxia. Newborns have smaller erythropoietin response to anemia than do adults because fetal hemoglobin, that is hemoglobin present at birth, carries up to the 30% of the more oxygen. The loss of the fetal hemoglobin due to insufficient erythropoietin production make the anemia worsen. This causes inadequate production of the erythropoietin. Therefore, newborn have a smaller erythropoietin responses to anemia than do adults. Now the maturation of the RBC. For the maturation of RBC, there are two vitamins are essential. One is the vitamin B12 and second one is the folic acid. All these are essential for the synthesis of the DNA, that is formation of the thymidine triphosphate, which is considered as a building block of the DNA. Now the lack of either vitamin B12 or folic acid causes first abnormal or diminished DNA, that is failure of the nuclear maturation and cell division. Second thing, causes the failure of the proliferation of the erythroblastic cells of the bone marrow. The lack of vitamin B12 and folic acid causes production of the larger than normal red blood cells, which is called as the macrocytes. The lack of vitamin B12 and folic acid causes the RBC with the irregular, large and oval shape instead of the usual concave disc. It also causes the failure of the maturation of the RBC during the process of erythropoiesis, and the form RBC have short life. Now the reticulocyte count. The rate of erythropoiesis is measured by the reticulocyte count. It then takes 1 to 2 days for the reticulocyte to lose the last vestige of endoplasmic reticulum and become RBCs. Normally, a little less than 1% of the RBC are replaced by the newcomer reticulocyte on a given day. Thus, reticulocyte account for about 0.5 to 1.5% of all RBC in normal blood sample. A low retic count in a person who is anemic might indicate a shortage of erythropoietin or an inability of the red bone marrow to respond erythropoietin, perhaps because of a nutritional deficiency or leukemia. A high retic count might indicate a good red bone marrow response to previous blood loss or to iron therapy in someone who had been iron deficient. Second, it could also point to illegal use of the epoetin alpha by an athlete. Now the next point, that is blood doping. Delivery of oxygen to the muscle is a rate limiting factor in muscular feast from weightlifting and running a marathon. As a result, increasing the oxygen carrying capacity of the blood, which enhances the athletic performance, particularly in their endurance event or the crucial event. The injection of the epoetin alpha, a drug used for the treatment of anemia, causes the increased RBC production, which ultimately leads to the increased oxygen carrying capacity of the blood. And because of this, the enhancement of the athletic performance. This is called as the blood doping or the artificial induced polycythemia. But blood doping or the artificial induced polycythemia increases the viscosity of the blood, which also result in the increased resistance to the blood flow. Because of this, the heart is in a difficult condition to pump the blood, and which increases the high blood pressure and ultimately increases the risk of stroke. There is an example during the 1980s, at least 15 competitive cyclists died from the heart attack or stroke linked to the suspected use of the epoetin alpha. Now the success of marathon runners from the Kenya. The average altitude throughout the Kenya highland is about 2000 meter, that is 6600 feet above the sea level, and other areas of the Kenya are even higher. The body increases the production of the red blood cells, which means that exercise greatly oxygenates the blood. When these runners compete in the Boston, for example, at an altitude just above the sea level, the bodies of these runners contains more erythrocyte than do the bodies of the competitors who train in the Boston. Therefore, a number of training camps have been established in Kenya and now attract the endurance athletics from all over the world. The altitude training greatly improves the fitness, endurance and performance, and this is called as the natural blood doping. This is a good example of the natural blood doping. Now we'll see some multiple choice questions. First question, healthy adult male have dash dash RBC per microliter of the blood. A option 5.4 million, B option 4.8 million, C option 6.4 million, D 3.4 million. The correct answer is 4.5 million. Now the second question, metabolic limit of cells hemoglobin forming process is dash dash gram in each 100 milliliters of the cells. A option 14, B option 15, C option 34 and D option 25. The correct answer is 34 gram. Third question, each RBC contains dash dash million hemoglobin molecules. A option 100, B 200, C 180 and D is the 280. That is correct answer is 280 millions. Fourth question, 70% of the CO2 is transported in the blood plasma from tissue cells to the lungs in the form of dash dash 1 A H2CO3, B HCO3 minus, C H plus and D H2O. The correct answer is HCO3 minus, that is bicarbonate ion. Maturation of RBC is regulated by A vitamin A, B vitamin B12, C folic acid and D both B and C. Correct answer is both B and C, that is vitamin B12 and folic acid. There are some long answer questions. Explain the physical characteristic of the hemoglobin. Second, explain in detail about the function of the RBC. Explain events involved in the life cycle of the RBCs. Why premature newborns exhibit anemia. References. First reference, Principle of Anatomy and Physiology by Gerard J. Tortora and Brian Derrickson, 13th edition. And second, Textbook of the Medical Physiology by Guyton and Hall, 11th edition. Thank you.